Hello, everybody, and good afternoon. Just kind of give it a couple of seconds while people are coming into the room. I don't know how quick people come through. Just a quick introduction of myself, just kind of while it's on screen and people are coming into the meetings. My name's Stuart Costello-Ferguson. I'm the training manager and quality manager at pathology in the Queen Elizabeth Hospital in Gateshead, up in the north of the country. And I'm just here to kind of host and help navigate through the session today. You're all here on sort of a healthcare science discovery session, learning about the professions and what healthcare science is. So a big welcome and thank you for coming along this afternoon. Will be, next slide, please, Paul. So session objectives this afternoon. So we're here looking to see what healthcare science is, what those professions are, but more importantly, an opportunity to have ambassadors, people working within some of those professions within healthcare science, to come and talk to you about what it's like working in those healthcare science professions. And there will be opportunities for questions and answers, live Q&A at the end of the session once all the speakers are complete. And we are here, obviously, as we sort of say, to get an insight into the roles using science within the healthcare setting, and a little bit how to get into those careers. So a bit of a careers advice and a careers knowledge there to be able to come and work within the healthcare science sector. We will sort of see the use of scientific knowledge. Sorry, next slide, please. So what is healthcare science? As you can sort of see on screen, it's the use of scientific knowledge, skills and equipment to improve the diagnosis, treatment and quality of life for patients. So what you'll sort of hear from this session today is that there's a lot of problem solving skills and analytical skills from the presenters across a variety of professions here to talk to you today. And those are kind of key skills for any healthcare scientist across any of those professions. But we'll also sort of see that there's a high level of sort of use of technology across those skills and across those professions, and attention to detail, which is sort of required for working within that healthcare science profession in that sector. Next slide, please. So within healthcare science, it's a sort of a broad term of professions. Right there in the middle, we can sort of see that there's over 50 specialisms sitting within that healthcare science sector. We can split those into kind of 4 categories really, and on screen there's just some examples of a few within each of those sectors. We've got from top left the pathology sciences, that work in sort of with, say, sort of biology and chemistry predominantly, and you might also see that otherwise known as kind of sort of the life sciences part of healthcare science. Moving around to the right, we've got physiological sciences, which are more kind of sort of patient-facing sciences. We've got medical physics and clinical engineering in the bottom right, sort of more sort of physics focused, I suppose, portion of healthcare sciences, and bioinformatics and genetics, who sort of writing programs and analysing genetic data. So a broad variety that covers kind of all sectors of science within that healthcare sector. So lots of options to choose from. So just before we kind of move on and meet our presenters that are here to talk to you today, we've just got a quick question. So just sort of test a little bit of knowledge there. We've got a poll that you can sort of click into right on screen there, but healthcare scientists have a role in the diagnosis of illness. What proportion of patients do they help to diagnose? Is it 30%, 50%, 60%, or 80% of patients? We'll just give you a few seconds just to pop your answers in. I don't know, Paul, does it automatically calculate, or do we? Yeah, so the answer we've got there is 80%. So quite a very high proportion of those patients coming through for sort of healthcare in the healthcare setting has some involvement with healthcare scientists as part of their diagnostic. So a large proportion of that patient experience. So without further ado, after that question there, we'll sort of move over to why you're here, really, to hear from those in the profession and sort of really see what it feels like to work within healthcare science. So the first of our job holders are the neurophysiologists. And we've got Melody, Scott and Catherine on the call here to talk about the amazing work they undertake for the NHS. If you do have any questions, and I'll sort of remind after sort of each session, if you do have any questions, we do have a live Q&A session at the end where you can get questions answered. Or if you have any questions that pop up through each of the talks, if you want to pop them in the chat, and we can keep an eye on those through the sessions and get them answered at the end as well. So without further ado, over to you three, Melody, Scott and Catherine. Thank you for coming. I'll let you talk about neurophysiology.
Thank you very much. So hello everybody. So yes, today we're just going to give you a brief overview of neurophysiology. So what we do, what we are and how we got into it. Next slide please. So in neurophysiology, we look at the function of your nervous system. So this includes your brain and your nerves. We do sometimes look at the muscles that those nerves supply as well. So you might have heard of a department called radiology, where they perform X-rays, MRIs, CTs. So these kind of tests look at the structure of different body parts. So we're more concerned with their function. So everybody's brain has little electrical signals it'll send as a way of telling different body parts what to do. So we look at the different kind of signals, either from your brain or from the nerves, and see how they're working and if they're doing what they should be. Next slide, please. So if you quite like problem solving, or just generally like getting the answers to things, then healthcare science is definitely something to consider. And if you like a bit of a clinical twist and the chance for a patient-facing role, then yes, neurophysiology would tick those boxes. So if you think of somebody with a set of symptoms as a bit of a problem-solving exercise, and the aim is to get to the solution, which would be the diagnosis and then possible treatment, your healthcare sciences are those pieces of the puzzle in between that will get you from problem to solution. Next slide, please. So we do a range of different nerve test. We essentially put small electrical signals into the nerves, so the nerves running down your arm or running down your legs, and then measure how much of that signal gets through. If there's any delay in that signal or not enough of the signal gets through, then that can show us that maybe that nerve is trapped or damaged somewhere along that route. We're trying to mimic the electrical signals that your brain will send normally. So if we stimulate a nerve that's supplying a muscle, we'll make that muscle twitch, as you've seen in that video. Next slide, please. Another test we do is called an EEG. So it's a brainwave test. So we're looking at the small electrical signals that your brain is constantly sending. So these brainwaves will look quite different depending on how old you are, or if you're awake or if you're asleep. So for example, a baby's brainwaves are going to be quite slow because the brain's still got quite a lot of developing to do. Then as you grow up, your brainwaves will start to speed up, and as you get older, they'll start to slow down again. That is perfectly normal. But sometimes your brain can get a little bit muddled and then send the wrong messages to different body parts, or maybe the entire body at the same time. This can cause body parts to do something that they might not be meant to do. This can lead to something called a seizure. And some people might be diagnosed with something called epilepsy, which is a seizure disorder. Next slide, please. For a bit of context, this is an example of a normal adult's EEG. It is lots of wiggly lines, but from this we can see how fast or how slow your brain waves are, what kind of shape they are, and if it's symmetrical, so are both sides of your brain doing the same thing. We also do a kind of crude heart rate at the bottom there, so you'll see that as the ECG. Each one of these wiggly lines corresponds to a different part of your brain, so we can look at each part a bit more closely. So for example, your frontal lobe or maybe your temporal lobe. We do this because sometimes a specific part of your brain can misbehave, and this might cause what we call a focal change and focal symptoms. An example of this would be if your temporal lobe misbehaves, then you might get a funny taste in your mouth, and that's because that's where taste is processed. Next slide, please. So this is an example of the whole brain misfiring at once. So you can see each of these wiggly lines is doing the same thing. It's an example of an absent seizure, and it causes somebody to go quite absent. So they might stop mid-sentence or mid-task, be quite still and become unresponsive while their brain is doing this, and then they'll go back to normal when it stops. Depending on what kind of symptoms you get and what kind of seizure you have, the EEG can look very different, and you can imagine there's quite a lot of different symptoms out there. Next slide, please. So we had quite an interesting case not so long ago. So somebody had been found outside wandering the streets, quite confused, not really able to speak properly. So they were brought into hospital by ambulance. The doctors noticed that she was twitching, so gave her some anti-seizure medication, and this did stop the twitching. But unfortunately, 24 hours later, she still wasn't speaking properly. She was still quite confused, and to be honest, they had no idea who she was. So we were asked to come up and do an EEG on one of the inpatient wards. So we put the EEG on, and we were there to try and confirm if she was having what we would call a silent seizure. So these are essentially quite invisible. So there's no obvious twitching or jerking, but somebody might just be a little bit more drowsy than normal, so a bit more sleepy, or maybe just a bit more confused. So once we switched the EEG on, we did confirm that she was actually having continuous silent seizures. So her brain was seizing the entire time. While we were recording, we got the doctors to administer some stronger anti-seizure medications, and within minutes we had the EEG back to normal, and the patient was able to sit up, converse normally, confirm who she was, and then we could find her on the hospital system. So EEG is quite interestingly the only test that can confirm and diagnose that kind of seizure. I'm going to pass you on now to Catherine, who's going to talk a little bit about how she came into neurophysiology.
Next slide, please. So my name's Catherine. I've been working at James Cook in neurophysiology for about 3.5 years now. I got into this after I finished my levels in 2021. So I did geography, biology and psychology. And then following that, I had just applied to university to go and study biological sciences. I didn't really know what I wanted to do. Felt like I had to apply to uni because everyone around me was. And at the time, I knew nothing about apprenticeships or being an apprentice, or that there were options like this available to me. Next slide, please. I then had a friend send me a link to a job ad on Indeed, and I then just applied blind, not knowing what neurophysiology was or what a degree apprenticeship was, and managed to land the job, which then led me to the next 3.5 years of my life. Applying for an apprenticeship is very different to applying for university. It's a typical kind of job. You'll need a CV, be interviewed, get hired by the department manager, and then they will put you on the university course. These are on things like Indeed and NHS jobs. If you just have a good look there, there's loads about in all sorts of different things. Next slide, please. So I then spent 3 years doing my degree. It was a very typical kind of, you work whilst you're doing it. And then I qualified last June in 2024 as a neurophysiologist. Next slide, please. So at first, having gone straight from my A-levels into this, all my friends had gone off to university, and I was watching them go out, have a really good time, whilst I was stuck going back and forth to work. And for a little while, I did really think I'd made the wrong decision. But eventually, I started being able to do other things. I managed to travel. I went to the likes of Thailand and Iceland because I'd been working. And that is not something I would have been able to afford to do had I gone to university and been a student. So there's lots of different pros to doing apprenticeships that you might not directly think of without being aware of what they are or that they're around. So if you're someone that wants to do kind of practical work whilst also still having a really good personal life, I'd definitely look at apprenticeships as an option for you. And I will pass on to Scott, who will tell you about his way into neurophysiology.
My name's Scott. I've been at neurophysiology for almost 4 years now. The way I got into it was a little bit different to Catherine. So where she came straight from A-levels, I already had a degree. So what I did following school is I had no idea what to do with myself. I was told, if you don't know, just do something you enjoy. And I enjoyed doing the science. So I went to college and did applied science. And then I went to university and did physiology for 3 years. But following graduation, I got to the end of it and realised, what do I do with myself now? There was nothing really waiting for me at the end of it. Go to the next slide, please. So I came back from uni, and I was doing lots of little jobs, but I really wanted something that said physiology in it, to kind of justify like the time I'd spent getting the degree and the student debt. Eventually, I came across neurophysiology on NHS jobs. I revised for it and I managed to get in. So this was a degree apprenticeship where we'd work 4 days a week and would have 1 day to do lectures and studying and coursework, all that kind of thing. But this was great because it essentially meant we got a degree while having a full salary and a career waiting for you at the end of it. So if I'd known about this before going to uni the first time, I definitely would have chosen this instead and saved myself the time and the money. If we go to the next slide, please. So as well as like having a degree with a career waiting for you at the end of it, it's also really good because it gives you the hands-on experience you wouldn't necessarily get with university. It's also really good for your personal development. So for me, it's made me much more confident and proactive. But it's sort of all to do with how much interaction you get with people. So I used to be quite quiet and shy before coming here, but through talking to all these different patients every day from like all different ages, all different backgrounds, and all the different staff members we have to come into contact with, that's helped me a lot. And at the end of it, I think an apprenticeship is a very good opportunity, and I would highly recommend it if you're considering university. That's it from us. We'll go to the next slide. Thank you very much for having us. Let you move on.
Thank you very much, E3. Really interesting. Sort of my background is in pathology. I'm a biomedical scientist, so it's really nice to sort of peek behind the curtain of other healthcare science professions and stuff. So sort of really good sort of seeing what you guys do and kind of how you've got into that role. And similarly, kind of my background was falling into biomedical science as well. So it's seems to be a common thing of finding it out after the fact, doesn't it? So as I say, if there's any questions in terms of neurophysiology, put them in the chat box there, or you'll have opportunities at the end of the session. So that was kind of very much a patient-facing role. So we're going to move on to sort of presenters for genomics and bioinformatics. So we've got Sophie, Hannah, and Thomas waiting to talk to you, although maybe not so much of a patient-facing role. Patient-facing role, very much directly helping with the patients, providing information to be able to target treatments specific to patients, which has been sort of revolutionary in terms of the treatment that we can give to patients. So without again further ado, I'll pass over to the three of you to go into more detail. Thank you very much.
Thank you. So just to say hi, my name is Hannah. I'm a trainee clinical scientist, and I've been working in genetics for 14 years, working my way from an entry-level medical laboratory assistant to a genetic technologist and finally a trainee clinical scientist. Tom and Sophie are also trainees, but we all work in a genetics laboratory. We're going to tell you about what we do for our work on a day-to-day basis. Just to mention, when we say genetics, you might hear people call it genomics, but they essentially mean the same thing. So just to sort of describe how your sample might get into our laboratory, like most of you will have been to the doctors when you're poorly, and some might have had blood tests taken. So if your doctor thinks you have an illness that has been caused by genetic changes, then they will send you to have blood taken. This blood will then arrive at our laboratory for testing, and the specialists who work in our lab are called genetic technologists, and there's also clinical scientists. Next slide, please. So everyone here is made of cells, and in every cell are chromosomes, which are these wiggly, stripy things we see here. And these are made up of DNA, which, as you can see on the left, is what codes us to become us. So we have 22 pairs of chromosomes, all are labelled 1 to 22, and 2 of our chromosomes are our sex chromosomes. Our DNA determines everything about us, like hair colour, how tall we are, but also whether we're likely to develop diseases. And we do tests in our lab on our chromosomes and DNA to look for these diseases that we are either born with, such as Down syndrome, or ones we could develop, such as cancer. Sophie and Tom are now going to talk a little bit more in depth about the work we do in the lab every day and how we got to where we are.
Next slide, please. Next slide again, thank you. So, hi everyone, my name's Sophie and I'm just going to talk a bit about how I got into genetics and my career progression within it. Next slide, please. So I did an undergraduate degree in biomedical science at university, and when I was at school, my favourite subject was biology. And I knew that I really enjoyed the human side of biology, which is why I picked biomedical science over just a general biology degree. But I was still unsure of exactly what I wanted to do in the future. And so biomedical science has lots of different avenues you can go down, which are shown on the slide. And through the course, you learn all these different subjects, and it's a great way to figure out what subject you might want to specialise in. And these are all careers that are working behind the scenes in labs within hospitals. And although you've probably never seen them, they are there. And so during my degree, I did a placement year in the NHS within genetics, which is where I became really captivated with genetics. And so after university, I got a job as a genetic technologist to pursue my career in genetics. And genetic testing is used to identify changes in our DNA sequence or chromosome structure. And this is helpful for the confirmation of a diagnosis that is suspected by a doctor, or for prognosis, which is the expected development of a disease, such as will it improve or worsen, and treatment as well, because we can use genetic testing to determine what treatments will work for different people. Next slide, please. So as a genetic technologist, the role is to perform tests in a lab on patient DNA samples to help determine a diagnosis for that patient. It's a very hands-on job, like many specialisms within biomedical science. So if you don't like the idea of being sat behind a desk all day, it's a good job to suit you. And genetic technologists perform many different tests. And so here we have an example of just one of the tests that they carry out called FISH, which is a technique that uses fluorescent tags which attach to patient DNA so that we can see specific genes to identify if there are any changes in those genes. And if a change is found, that can help us to determine what treatment a doctor should give to that patient. So on the left, we have me making some fish slides and also an image of me using a fluorescent microscope, which has to be used in a dark room so that you can see the fluorescent tags. Next slide, please. So this is what we're looking at when we do FISH, and we use this in cancer diagnosis. So when a patient has cancer, we can look for changes in the DNA of the cancer cells in their body, which might mean they will or won't respond to certain treatments. So we get a cell and we add probes, which makes the genes within DNA glow so that we can see them down a microscope. And at the top, we can see what a normal cell should look like. So you've got the blue circle, which is the nucleus within the cell, and that's full of your DNA. So we see 2 red signals and 2 green signals, which are the glowing genes. And the cells on the bottom show changes that occur in cancer cells. So on the left, we have a deletion. One of the red signals has disappeared because the gene it is attached to has been deleted. In the middle, we see 2 fusion signals, and that's because in that cancer cell, the genes have stuck together. And so if a patient has a fusion or a deletion, they might be eligible for a specific treatment. And then finally, we see amplification where there are lots of green signals because that gene has greatly multiplied in the cancer cell. And this is associated with a poor outcome for the patient. So I really loved my job as a genetic technologist, and it was very hands-on, but I was not always seeing the end result of the testing because that's more the role of a clinical scientist in genetics. And so now I'm training to be a clinical scientist on the STP, which is a training scheme where I learn on the job and my workplace pay for my master's degree. And it's much more of a desk-based job than a genetic technologist is. But as scientists, we still work really closely with genetic technologists as I get to see the data that they produce. And then I have to analyse and problem solve using that data to determine why the patient is unwell and what treatment may work for them. And then we can pass this information on to doctors. So we also work closely with doctors. So some of the skills that are important in genetics are problem solving, communication, teamwork, and also compassion. Because although these roles are not patient-facing roles, every day we do get to see the impact we have on patient lives. And so it's a really rewarding career. And I'll pass over to Tom.
Cool. Thanks, Sophie. Hi, everyone. I'm Tom. I'm another trainee clinical scientist working in genomics/genetics. However, I'm a bioinformatician. So we're looking at more the computing side and looking at actually sequencing your DNA. Next slide, please. So whenever I say bioinformatics, I normally get a bit of a blank stare. So I like to break it down into these three kind of core areas. The first two, biology and statistics, are kind of shared by healthcare science in general. From a biology standpoint, we need to know what DNA is, what genes are, what disease we're looking at. It's really just all the context of what we're going to be looking into. Statistics is going from data to decisions. So we need to know what is this usually very big and complex data mean and how confident are we of our results. We don't want to give people wrong results where possible. The third area that makes us unique to other specialities is computer science. So as we were very nicely introduced at the start, we do programming, use algorithms and sort of logic-y stuff to turn these really big, complex, understandable data sets into actual usable stuff. Next slide, please. So as from a science background, not a clinical background, and I knew that I really enjoyed biology and coding, so I wanted to use both of those and do something that was a bit rewarding. I also liked variety and learning new things. I knew I could learn independently and problem solve for my degree, and those are things that I was really interested in doing more with my career. Bioinformatics was a really good opportunity for this. It's a really quickly progressing specialty. It's 40 years old, which seems really old, but in the grand scheme of things, it's very, very young. We're now looking at using things like AI and cloud computing, which is really just shaking up how we do everything. So I've always said that if someone's asking a question or, and you've just thought, I could Google that, or you should have Googled that, this is probably a good role for you because there may not be an expert on your team or your department for what you're looking at. You're going to have to go away, research, see if it's useful, and then implement it in your sort of day-to-day practice. Next slide, please. So a bit about what I do kind of day-to-day. As Hannah sort of introduced, you might go to the doctors, they'll take a blood sample, they'll send it to us. We prepare that and extract the DNA, put it in one of our machines, and then we get this kind of data off, which is, the A, T, C's, and G's you might have heard about that make up your DNA. We're reading those and putting them out as files. This doesn't mean anything to any of you or me, and shouldn't to anyone who doesn't have too much spare time. We get 1.5 billion of these letters per patient per sample. If you printed that on A4 in normal sort of font, that would be 50 meters tall, or about 15 stories, just for one sample. And we do 300 samples a week. So as you can imagine, that is a huge amount of data to try and do anything manually. A big part of my job is to turn this data into useful results and automating that where possible. So we want to take out that human component anywhere we can. So I managed to cut our processing of this data in half by automating it all. That cut about 2 days off our testing, which doesn't seem a lot, but when you're at home waiting for a test result, being able to get that result 2 days faster can mean everything. So although we're not patient facing, we're really kind of in the background, that doesn't mean we don't still have a really big impact on patients and their experience. Next slide, please. So as I said, we're a varied job. We're not just doing data. We also do bits of things like web development. So here's a web app that we developed in our lab and that I help maintain. And really its job is to let scientists and clinical staff all work together to analyze the data, interpret what it means, and then put it out into a nice report. Just make sure that everybody, make this sort of process as streamlined as possible. Next slide, please. So, this is my final slide. All the data goes through that app, and then we come out with these nice little reports. So we've gone from 50 meters of paper down to maybe 3 or 4 genetic changes that are either going to impact your disease, your treatment, or how things are going to go. And then we hand that back to sort of the clinical staff. So Hannah's just going to finish off a bit about how these results get back to you. Thank you for listening. Next slide.
Yeah, so thanks, Tom, and thanks, Sophie. So as you've seen from Tom and Sophie, once we've finished testing on the samples we receive, we then need to send reports to doctors and clinicians for them to deliver these results to patients. And another part of the genomics team is a group of people called genetic counsellors, and they have a really important job of delivering these results of the testing we have done to the patients in a way that is understood. Genetic counsellors have to be good at listening and communicating, interested in people and the sciences, and also have a lot of kindness and compassion for the patients they are delivering sometimes upsetting test results to. Genetic counsellors will guide and also advise patients on their conditions and also involve other family members if the condition can be passed on to others. They also explain any treatments that might be necessary for the patient. In order to become a genetic counsellor, you can do a degree and then apply for the scientist training programme in a similar way to Sophie and Tom. So one difference between the job Tom, Sophie and I do and the genetic counsellors is that they are patient facing. So if you prefer the sort of interaction with people, but also have sort of a genetic background and interest, then, that would be the job for you. Whereas we're very much, as has been said, working in the background. And whilst you might not see us clinical scientists, we are very much a part of a patient's journey to diagnosis and to treatment. Thanks for listening. Next slide, please.
Thank you very much, Hannah, Sophie, and Tom. Really good. Again, another profession that I don't know much about it, but it's really good to see the sort of broad diversity, even just within genomics itself and the professions that you can get into. Thank you very much. Again, just a reminder, any questions around sort of genomics, bioinformatics, pop them in the chat there, or you will have an opportunity at the end about sort of the live Q&A to ask the questions that you like. So again, following on from that, next up we've got David, who's the Chief Scientific Officer for Leeds Teaching Hospitals. It's the head of the healthcare science profession for the Trust, and his role provides clinical and professional advice and leads on the use of science in order to improve health and health outcomes. So yeah, again, without further ado, I'll hand over to David to tell you about his role. Thank you very much.
Thank you, Stuart. Good afternoon, everybody. As mentioned, my name is David Brettle. I'm the Chief Scientific Officer at Leeds Teaching Hospitals, and I think I'm the first Chief Scientific Officer in Leeds Teaching Hospitals, and probably in many trusts. There are other roles similar to mine, like lead scientists, or you'll hear titles like Director of Science, but I am the first Chief Scientific Officer. So, what does that mean? Well, you've just heard from colleagues who are potentially earlier career, starting out in their journey in healthcare science, and I'm at the other end. I'm at the end of my career. It sounds a bit sad, really, but there you go. So gives you a flavour, hopefully, hearing the perspectives from those who are early career and from me now at end of career. So what is a chief scientific officer? Well, you may have heard of a chief medical officer, a chief nurse, and other chief titles in organisations. And these are basically what they call C-level managers. We're supposed to be the most senior people in our professions. So I'm the most senior scientist in the hospital. It doesn't mean it make me the best scientist. Does it mean I know everything? That is not the case. But it means that I am responsible for science in the hospital. That includes the professional part. I am responsible for the workforce. There are, believe it or not, 1,300 healthcare scientists in Leeds. And to put that into context, I think there are around 4,000 nurses, around 2,000 doctors. So there's a lot of healthcare scientists, and we're often unknown and sort of not in the public eye, shall we say, but they're definitely there. The other side that I lead on, as Stuart mentioned, is the science side. So I am the lead scientist, I'm the chief scientist in the organisation. So often the organisation needs advice on science, on technology, on innovation. So one of the biggest parts of my role at the moment is leading on innovation, because innovation has that ability to transform what we're doing, to bring in new technologies into an organisation, such as genetics, bioinformatics, AI. All of those are hugely important technologies, and they need to be brought in safely, and they need to be managed in the organisation. My background, I'm a medical physicist, one of those segments on that careers pie chart you saw at the beginning. So most of my career I've been working with radiation. I've been doing X-ray safety, both staff and patients, but also magnetic resonance imaging, ultrasound, pretty much anything to do with images. Images is my thing. I've even done a bit of AI in the past, so that makes me sort of relevant, maybe not completely end of career. I've also had an interesting career in that I've been half doing the day job, which is being the medical physicist, but actually half doing research and innovation. So I've had loads of innovations. I've got loads of patents, loads of inventions that are being commercialized or have been commercialized. But the research side has very much been around imaging, image processing, and that data analysis role. Currently, I work in NPIC, which is a National Pathology Imaging Cooperative, which is basically leading on rolling out digital pathology across the UK. So those slides that you see under a microscope where you can look at the cells, it's basically digitizing those and making those available on a network across the UK. I absolutely passionate and love my research side, and particularly the innovation and inventing. Next slide, please. So that's what I am, that's who I am, what my role is, but what do I do? I thought I'd just go through my diary, basically, for the last few weeks and summarise what I do. These photos show me actually doing my job, which is getting out, meeting people. Don't do as much of that as I'd like to do, pressure on time and all that. I do give lectures. And yes, I even went to Disney World for work, which is a hard job, but somebody's got to do it. So I've had fantastic opportunities, which I'll tell you about some of those in a moment. So day-to-day job. Well, since the end of January, and don't get me wrong, I'm not perhaps typical. Like I said, I am probably one of a very few number of chief scientific officers in the NHS. But since January, I've literally been overseas twice. I've been to Vienna and I've been to Sweden, both for conferences, academic work. One was educational and one was pure research, which is great because I've never been to Sweden before, so that was really nice. I've had loads of senior leadership meetings, and trust me, these in a hospital is 24/7, so sometimes these meetings are very early. In fact, I have several meetings at 6 a.m, which is harsh. And every week I at least have one at 8 a.m. So that can be challenging because I'm a night person, not a morning person, that's for sure. I've also had loads of meetings around my inventions because they're coming to fruition. I've got one that's just being commercialised, which I'll tell you a little bit about in a moment. It's been launched actually in May, and another where we're having advanced discussions about commercialisation. So I've been doing lots of working out about what that commercialisation looks like. We had a major event in Leeds recently in our it's called our old medical school, which is basically a building we have that we're turning into a health technology centre. So we had 110 innovators, industry, clinical, we had politicians there. We even had Tracy Brabin, who is the mayor of West Yorkshire, came and joined us at that event. And that was absolutely brilliant to be in a room with so many innovators, so many people like-minded. A boring thing, but every week I meet my research team. It's not boring to me. It's one of the best days of the week because I get to hear about how the projects are going, how my team are doing, and to meet them. Again, this picture, the reason this picture here is not to show me out and about, it's to show the people, because it's all about people I work with, people who make the magic happen day to day. We've heard about some of our colleagues earlier on in this presentation, but these are others that do exactly the same. So I also, next slide please, sorry. So I also do some rather interesting things. Here I am in the photo. This is in Oslo in Norway, and I'm using HoloLenses to visualize tumours in a brain for preoperative planning. Because obviously the thing about innovation, you want to see the innovations get out there, and this is getting out there and seeing what they were doing in Norway. So again, part of my diary, I do lots of visits. Since January, I've had 3-4 visits, which includes 3 bishops and a philanthropist, can't say the word, but who happened to be a co-owner of a Premier League football club. So that was quite cool. I've never met an owner of a Premier League football club before. So far, this is my 3rd webinar this week, in fact, and one was for the Science Council, which is also celebrating British Science Week. And I think that webinar, which was on my life journey, how the ups and downs of research, I think that will be on YouTube soon. So if you're interested in hearing a bit more about the ups and downs of research, please go and check that one out. I gave an interview to a Dutch company, I think it was, working on a big European project we're involved with, and basically, again, an interview about my work and what we do, which was interesting. So that's sort of 6 days worth of, 6 weeks worth of my job, and it's pretty similar in that it's pretty diverse. I can be doing a different set of things every 6 weeks. I don't have a fixed job. I don't have a day-to-day routine. My job is to be available and to lead and to do strategy and to take forward. Career highlights. Thought you might like to hear about some of that highlights of my career. Next slide, please. So first of all, on the left there, that is without a doubt the career highlight for me. That was the project that I initiated called the Little LINAC. So a LINAC stands for Linear Accelerator, and this is a device that delivers radiation to tumours to kill the tumour. For patients with cancer, they will have that treatment over a number of sessions, and it's in a darkened bunker underground, which is scary for anybody, but it's particularly scary for children. So to help them with that treatment through play therapy, I developed this model brick kit, which we gave free or continue to give free to a child having radiotherapy for cancer in the UK, and it's gone worldwide as well. The other picture shows a little box there. That is actually a ventilator. We developed this from COVID, during and from COVID, and now this device is a low-cost, easy-to-use ventilator that is now out in Uganda. It's being clinically evaluated. It's just being used in another trial to look at end of life and paediatrics, and we're hoping that they're going to start manufacturing them in Uganda. And this is around frugal innovation, giving innovation to the world, basically, to areas where they have low or no technology. I forgot to mention I actually have a little INAC with me if anybody would like to have a little look. Hopefully you can see the pictures there. You can see how it's supposed to articulate the same way that a real machine does, so a child can play with that if they need to. I forgot I had that. I've been at Houses of Parliament several times, which is really nice, having dinner in there. And I also get to meet occasionally politicians, royalty, etc. And I even got to meet Dr. Ranj. If you know Dr. Ranj, he led the TV programme Get Well Soon and was a really interesting person. So I'm not sure that's a highlight, but certainly it's interesting. And I've travelled a lot. People often ask me if I get to travel in my job. I may be unusual because I have both a research, professional and work related need to travel sometimes, but I've just jotted down the countries I can remember I've been to, and it's massive. It's Brazil, Thailand, America, specifically Orlando, San Francisco, St. Louis, Chicago, Washington, Boston, San Diego, Las Vegas. I've been to Turkey, Greece, Sweden, Finland, Germany, Holland, Belgium, Austria, Norway, France, Switzerland, Malta and India. Oh, and in May, I go to Singapore and China, and that's to launch this new product. So it's pretty cool, really. You know, I've had a fantastic career. Next slide, please. So you're probably sitting there thinking, well, that's all right for you, David. You've had it lucky. You've probably got rich parents, brainy, went to Oxford, Cambridge, maybe, privileged, whatever. No, that's not the case. Not the case at all. I was brought up on a council estate in Birmingham, near Birmingham. I had a very difficult childhood. I was actually invited to leave the Cubs, which actually means I was thrown out of the Cubs. I was invited to leave the sixth form first time round. I've cleaned the streets. I joined the Royal Air Force. I was also invited to leave. I went home eventually and was unemployed, did my A-levels again. And that's when I discovered physics. Physics has been my savior because I can do physics and not a lot else. So I am not some brainy person with privileged background. I just found my passion, and that has given me a career and could for you too. First in my family to get a degree. I'm probably dyslexic or have ADHD, but that didn't exist when I was a kid. I was just a troublemaker who was underachieving, and that was hard. And it is hard, and I won't pretend it's easy. Sometimes you have to work harder, and that's okay. And you have to work out your tricks as well. You know, do you use dictation, voice notes, YouTube videos to give you extra information? Whatever you need, my advice is find those tricks that work for you, and also find that secret talent, because we all have a secret talent. And my secret talent is inventing things. And who would have known it's also in making little Lego models as well. So that's my secret talent. So I've had a fantastic career. I've done amazing things, and it's all through a love of physics. It's all through healthcare science. But best of all, for me anyway, is that I've helped, and I hope to continue to help patients on the way. So my final slide, next slide, please. It's not all glamorous. Here's me biting down on an ice block whilst being imaged with a thermal imaging camera to look at teeth vitality. But hopefully I've given you an insight into my role, and I'm really happy to take any questions in the Q&A session. Thank you.
Thank you very much, David. Yeah, really interested in what our career and what some experiences, by the sounds of it, that you've had the opportunity to see. And I think kind of that advice piece that you had at the end there really resonated about sort of finding your passion there, finding what makes you tick, because that's where you're going to work towards, isn't it? Make you enjoy your job and everything. So yeah, so that's the last of our talkers. Thank you very much for everyone that's contributed this afternoon. They are all still on the call, so we do have a Q&A session here and it will kind of open those questions out to the panel and those on the call. I guess kind of first off, we've just having a look through the chat, we've got a few questions kind of been populating through the chat box here. So I'll start from the top and I'll work the way down. If you've got any kind of live questions, if anyone wants to come on mic to ask those questions, feel free to raise hands and we'll kind of get through to those as well. Right, so the first question that I can see in the chat, kind of top to bottom, from a Niarika Fukan. Apologies if I've mispronounce that. But we've got, what careers would you advise for an international doctor with clinical and pathological lab experience, especially oncology, to take up in the UK to progress towards a cancer research career? Do we have any answers? Because I'm not sure. I'm not sure in terms of a research career, unfortunately. We can always take these questions and kind of see if we can find answers and get answers back to you if we can't answer them in the call today.
Sorry, Stuart, can you repeat the question, please? I might be able to answer that one.
Oh yeah. So what careers would you advise for an international doctor with clinical and pathological lab experience, especially oncology, to take up in the UK to progress towards a cancer research career?
Okay, so obviously these are very sort of person specific and circumstance specific questions, and so I'm going to give a sort of general answer, I guess, but the advice I would give is to reach out to professional bodies. So you know, there are many professional bodies in the UK who often advise on you know, specific translation paths, for instance, from other countries or other qualifications or equivalent routes. So my advice is always, in these circumstances, reach out to professional bodies and get them to give you advice and support on that.
Cool. Thank you very much, David. Next question we've got is from an L Castle. Is it acceptable to do a degree apprenticeship following a degree in a similar subject? I'll sort of quickly answer from my perspective that the easy answer is probably yes. I think the apprenticeship regulations and rules and stuff is that as long as there's significant learning that will be found on the new apprenticeship, then yes, you will be able to progress onto that apprenticeship. And in some circumstances, if you've done a similar subject and some of those modules from the previous degree kind of cross into the degree that you'll be doing on the apprenticeship, you can sometimes kind of APEL some of those modules depending on the apprenticeship provider and the degree provider. So you can actually kind of move that prior learning into the new degree, and so you kind of effectively top up to the new degree. Certainly that's the case within biomedical science, in my experience. I'll kind of open out to the rest of the panel to sort of see if there's any other answers or anything different to that answer.
Oh, well, I did a degree in physiology before doing neurophysiology, and it worked for me. If anything, it probably made my CV stand out a bit more. But I think so long as you have the relevant sort of background education, you can apply for it, yeah.
Thank you very much, Scott. So next one we've got is from a Chloe Huxtable. Can you go into neurophysiology without doing chemistry A level?
So yeah, absolutely. Like I said in my little bit, I did biology, geography, and psychology. So none of those were chemistry. But the main thing that we've kind of just discussed when we saw the question come in was, if you have kind of one typical science, so biology, geography, physics, and then the other two A levels just need to be relevant. So soft sciences, sports science, psychology, anything else that is just a good A level to do would get you into the uni. Each uni has different requirements, but typically 3 A levels with one science subject would be fine.
I didn't even do A levels. I did an A level equivalent, which was applied science, which was also relevant.
Cool. Thank you very much. Next question we've got from a Megan Roker. Is there an alternative to the STP to become a clinical scientist as a postgraduate, specifically in neurophysiology?
So we were also kind of discussing this as well. So I did the STP to get into neurophysiology. Once you're in the job, once you get to a certain level, there's not much difference between kind of me as a clinical scientist and what I do and some of my colleagues. So if you came into neurophysiology, say through an apprenticeship route, and then kind of internal training would get you to a certain level that you could then apply for like an equivalence of a clinical scientist within neurophysiology, where you do the same role.
Cool. Thank you very much. We've got Aisha Khan. Did you have to build out projects to land your first bioinformatics role?
Yeah, that's the one. So I taught myself to code on one of these online coding platforms. Just demonstrating that you have some coding knowledge is usually enough, and a bit of knowledge and interest in a particular area you're going into is usually enough to land a first role.
Brilliant. Thank you very much. Oh, I've lost. There we go. Got a question from Mavish. Would genetic technologist role require IBMS portfolio completion with a biomedical science degree?
Not at the moment. So a lot of people that I worked with as a GT didn't have an IBMS portfolio. Whether that will change in the future, I'm not sure. But they did have the opportunity to get the portfolio while in work as well. But yeah, it's not essential.
Okay, thank you. Niharika Pukan, could you elaborate more on the job role of STP cancer genomics?
So I am currently doing the equivalence route in cancer genomics. So there's 2 routes into becoming specifically a cancer clinical scientist. So one is the STP. But both end up with the same sort of job role. So on a day-to-day basis as the equivalence, I've got my own service that I run, which I basically have helped develop within my department certain bits of testing. That was part of a research project that I've done, and that's kind of ongoing. And I lead that service, specifically in uveal melanoma samples. That's a major part of like my role. As a trainee, you take on the booking in of samples. So when we receive the samples in the lab, we have to, as clinical scientists, determine from the referral form what pathway the samples need to take. So whether they go for extraction of DNA or whether they go for culturing of cells and things like that. So that's again a major part of our role, alongside analysis. So we have lots of different types of analysis that we do. There's whole genome sequencing, which is looking at a person's cancer in its entirety. I don't know if people have heard of the 100,000 Genome Project, but it's all that kind of looking at the DNA of a cancer sample as a whole, to look for sort of novel genetic changes. And also we have sort of NGS, which is another type of sequencing, but it's not the whole like the whole genome sequencing. So we have loads of different analysis that we have to do. And yeah, as part of my equivalence, I have to create a portfolio, which I then go through, put forward for once I've completed, and then I complete like an interview of either. Whereas the STP, they have a separate sort of university based part of their learning. So whereas mine is all based in work whilst I'm working and I'm learning the job, the STP focuses on sort of university, doing a master's, a research project, and then they come into our department and work alongside us to learn the different areas. So yeah, I hope that's helped.
Brilliant, thank you very much, Hannah. Really informative, hopefully. Yeah. So next question we've got is Kaleem Azar. Looks like a couple of questions about the STP program in particular. limited knowledge about the resources we can refer to while preparing. So I guess kind of where they get those resources from when applying for the STP, I think. Question whether or not there's the opportunity for internal recommendations for the programme if you're already an NHS employee. And then also one there about international recruitment of cardiac physiologists. And some query about the permanent closure of recruitments. I'm not sure about any of that, so I'll put it out to those on STP programs.
I can answer the one on the STP. So on the National Healthcare Science, I can't remember the exact name, but on the School for Healthcare Science, there's absolutely loads of resources for the STP. And they do give you like information about the SJT and some practice questions as well. In terms of being an NHS employee, so you can do an in-service route for the STP, but in terms of it being the easy route, I wouldn't necessarily. It's probably easier in terms of competition, but you're still going to have to get a job within a lab, and then there's also other people within that lab who will also be looking for the same opportunity. So it's still highly competitive within the workplace. Also, different labs offer different opportunities. So I know that some people that I work with on the STP, they know people who have gone through the equivalence route, which is what Hannah is doing. So you're not necessarily going to get onto the STP because a lot of labs will maybe push people through the equivalence route as well, just because they've got then someone who's working like directly on the job. So I hope that answered the question.
Thank you. And I don't think we've got any cardiac physiologists on the call or that been on the panel, so I'm not sure we have anyone to answer the last question there. Correct me if I'm wrong. No, apologies. So we've got a question from Mavish. What would be the best way to get into a biomedical science role? Degrees accredited but require IBMS portfolio to be a biomedical scientist. What are some of the routes? So certainly I'm a biomedical scientist. So from my perspective, I get asked this question quite a lot because we do get quite a lot of people that come through university, get their degree, and unfortunately aren't able to get placement in order to do their portfolio. So kind of my, I suppose, piece of advice would be looking for like trainee BMS positions. So in that you would come in as a trainee band 5 and to take your portfolio. Upon completion, you would be then moved into a registered BMS band 5 position. Alternatively, if you look for sort of band 4 positions, some labs will support doing that RBMS portfolio in a band 4 associate practitioner position. Upon completion of that portfolio, you're then able to sort of apply for registered band 5 positions. You won't kind of automatically be put in, you'll go through the same recruitment procedure on sort of NHS jobs. Alternatively, to kind of, for coining a phrase, sort of get your foot in the door, apply at sort of the band 2 or band 3 specimen reception, sort of medical lab assistant roles, kind of get yourself into the lab, and then sort of internally you can kind of be first up to sort of know about vacancies coming through, I suppose, really, and get some of your face known in your local lab to then kind of be able to go into one of those sort of 2 employment positions to then be able to undertake your portfolio. Alternatively, they might be able to then sort of the apprenticeship pathway might be available to you once you're employed within an NHS lab. They might be able to support you through sort of the degree. Well, I suppose you've done the degree, I guess, haven't you? But yeah, no, scrap that last apprenticeship bit, you've done the degree. I'll open it back out to everyone else and sort of see if there's any other alternative answers. No. Okay. Samuel Adegbola, an international biomedical scientist with a master's degree in molecular biology, specialising in bioinformatics. How can I enter it into the UK job market?
I mean, I guess I'll try answer a bit of that. Not as someone who's domestic. Yeah, I think just having, for bioinformatics, having a little bit of a portfolio will help. So having some projects we worked on, be able to demonstrate that experience, I think is really valuable. As I said, a bit of independent and problem solving. If you can demonstrate those skills, I think will put you in pretty good stead for bioinformatics roles. I think, yeah, that's all I can say really.
Well, thank you very much. We've got a question from B. Could anyone please advise about a career in microbiology? So I guess from a biomedical scientist perspective, microbiology sits within sort of the pathology disciplines, the biomedical science disciplines. So it'll be the same route of entry of going for biomedical science degree or applied biomedical science degree, with your sort of 3 A levels or sort of the required UCAS points on entry to that sort of degree program. Making sure that it's an IBMS accredited biomedical science program. There are non-accredited ones, and if you go through the non-accredited, you won't be able to become a registered biomedical scientist. So just make sure it's IBMS accredited when applying. Once on completing on that degree and your portfolio and able to get the registration as a biomedical scientist, you can sort of apply for jobs within microbiology. So kind of the registration portfolio is generic, and you can choose which discipline really that you want to go into and what interests you. So yeah, sort of microbiology is all about kind of the identification, the growth and identification and treatment plans for sort of bacteria and viral infections. I'll open it back out again and see if there's anything that anyone else wants to add. Nope. We've got one from Alice Lamparelli. Is there anywhere I can go to read further into those innovations? That might be towards yourself, David, potentially.
Gosh, just search me online and you'll find many of them mentioned. Particularly the little linac, just put little linac in. But the most of the inventions now are around pathology and imaging. So if you find me on LinkedIn, you may find a lot of that coming up in there as well. So it's probably the best way, to be quite honest.
Brilliant, thank you very much. We've got Gethin Slatter, currently biological scientist, undergraduate, interested in becoming a clinical scientist through the STP, but feel like I haven't got enough experience for my CV. What would you recommend for those maybe that are on the STP or the trainee STPs? Any advice for getting more experience?
I mean, I was just going to say, just even applying for, I suppose it depends what you're wanting in terms of your experience or how long, but I mean, applying for sort of medical laboratory assistant jobs, you know, lower down jobs, but more entry level jobs that don't require like the degree or anything like that. That's quite a good route in. It's the way I started out, and I know I'm doing the equivalents, not the STP, but I know I've interviewed lots of MLAs and AGTs and GTs as well, so assistant genetic technologists and genetic technologists who have all gone on to get onto the STP, and it's a really good way of showing like your dedication to, certainly in genetics, and I'm sure it would in other laboratories as well, but just it shows your dedication to genetics, but also shows you understand the workings of the laboratory, so it stands you in really good stead for, you know, the STP interviews and things like that, because you've already got like a really good background. So I mean, that's kind of where I would say would be quite a good starting point, I think.
And I think we've just had some useful links kind of put up on the screen there. So kind of all these questions about how kind of get into the careers and sort of the additional experience will kind of be fed into those links. So definitely a starting point for looking through all of these as well, for how to entry onto the relevant qualifications to get into the professions that you're interested in. We've got a question from Odupe Olorunfemi. Doing a PhD in automating clinical coding process. A question specifically for Tom: which career path can they focus on?
So yeah, I just want to think, because this is slightly an evolving specialism in NHS. would be clinical informatics, I think, is technically. So it's kind of related to bioinformatics, but more around management of like data and sort of business analytics, things like that. So that's probably more of the route you could go down. I think generally, I work a little bit with our general informatics team, which is more sort of project focused. So maybe keep an eye out for things like that if that's what you want to get into. Yeah, I think they're probably the best choices.
Well, thank you. Question from Priya. Some advice for a Year 12 student aiming for a biochemistry degree or career. What jobs would be available for someone with that degree? So in terms of kind of healthcare science, you can go into clinical biochemistry. So in order to get that, that's kind of my background and the route that I've taken. So it would be looking at sort of a biomedical science degree. So again, looking at EU CAS points for access into applied biomedical science degree, completing on that, and then sort of looking for jobs at the end of that or placements if you're doing an applied biomedical science degree, specifically within sort of clinical biochemistry departments, which generally kind of all trusts really have, or majority of trusts have, the clinical biochemistry as part of their routine pathology labs. So having a look for experience in there. Again, I'll open out if biochemistry allows you on to any of the other professions that have been spoken to today.
Yeah, I started as a biochemist, so it's a good underpinning for a lot of diagnostic testing. Biochemistry, just learning about DNA, amino acids, proteins, all that sort of stuff. So yeah, it sets you up for quite a broad range of stuff, I'd imagine, health science.
Yeah, it's a good starting point. We've got a question from Andrew Wallum. What proportion of degree apprenticeships are open to A-level leavers and what proportion just for internal applicants? Is there any tie-in with T-levels in West Cumbria? So my understanding with apprenticeships is you need to be in an employed position. So you'd have to find a job band 2, 3, 4 banding positions in an NHS lab. Once you're in an employed position, they're able to then put you through on the apprenticeship program, because it's sort of the funding rules behind being able to access the apprenticeship programs. I'm not sure on direct entry apprentices in some healthcare science programs. So when I say direct entry, it means that you can directly apply onto an apprenticeship program, having not sort of prior been employed as an NHS employee. So yeah, you'd probably need to look for employment first and then sort of have that support to move on to the apprenticeship program. And then T-levels, I guess T-levels are the equivalents to A-levels. So it's checking with your local or the university that you're looking to apply to make sure that T-levels are an acceptable qualification for accessing onto that particular degree route that you're looking for in that program that you're looking for. My understanding that it is becoming more sort of acceptable and equivalent to being able to access. Again, I'll just open out just in case I've got my understanding wrong and if someone else on the call is wanting to challenge that. Nope. I've got a question from Irfan Khan about sponsorship for employment in the field of biomedical science and medicine. I don't know anything about sponsorship, unfortunately, so does anyone else on the call have any experience? Nope. Apologies. But I would think sort of if you have a look at these health careers or sort of the HCPC websites, you might find more information there about sponsorship. Why is it difficult to get employed as biomedical scientist or health science assistant as an international person? As far as I'm aware, as long as kind of you get in the sort of the experience in and the sort of the prerequisites for employment, you should be under just the same expectations really for employment to any of the NHS professions, any of the healthcare science professions. So I guess it kind of comes back to getting that experience, maybe those answers that we've sort of made before, and bringing that to kind of the interview panels really and into your application forms. Just have a quick scroll through. Oh, Abe, I'd like to ask David about his career path. Starting out as a lab scientist, how do you transition into healthcare management? What's the balance of technical expertise with management responsibilities? What would you have done differently earlier in your career that you would have made it easier for you?
So the transition from operational day-to-day, I guess, into management is one, taking on those sort of early stage leadership roles, leading a small team and getting that sort of grassroots experience. But to make the big jump, I guess, into being a proper manager, leading a service, particularly for me, was the breakthrough was doing a postgraduate certificate in health and social care management, and that allowed me, it's literally one of the requirements for a senior leader role to have a formal management qualification. So that's definitely the key one. I think it's really important as well as a senior leader in science to be credible. So keeping up that research profile is important as well, because when you get those job descriptions, they will basically say you have to have a track record, and that's how you demonstrate your track record. But also, how would I have done things differently? I guess would basically move more. There are jobs out there, but if you're willing to move, then you get an ability to get promotion a bit faster. But I couldn't move because of family, so I had to wait for jobs to become available. So that can slow things down a little bit. But definitely, if you want to move into management, I'd recommend it for anybody anyway. A leadership management course like a postgraduate certificate in health and social care management is fantastic. Really useful skills.
Brill, thank you very much. Oh, Hannah.
Yeah, hi. Sorry, I've just been looking into whether it's still relevant. I think Andrew Wallum mentioned about the proportion of degree apprenticeships open to A-level leavers. I know that in our department in Sheffield, we used to take on level 2 like healthcare assistants as apprentices in genetics. These things are still available. They're not specifically in Sheffield anymore, but just look out there for the healthcare scientist assistants and associates. That's the kind of thing. But there are level 2 and level 4 healthcare science apprenticeships available, and for the level 2 all you need is A levels as a point of entry. So sorry to interrupt.
Cool, thank you very much both. Unfortunately, we are going to have to wrap up the webinar. By the looks of it, I think Paul has put an email through email contact at england.nshcs@nhs.net to forward any emails that have unfortunately we've not been able to answer today, and they'll be able to forward through with some answers or kind of signpost to some relevant links. But I suppose on behalf of the panel and those speakers, thank you for everyone joining on this afternoon. I hope you found it useful, and thank you very much to everyone that has spoken and given your expertise and your experience. It's been really informative. Have a good afternoon, everyone. Thank you.